Good morning.
Morning.
Hey, Beth.
Hi.
How's it going, lady?
Good. You're the host of this meeting?
Yes.
Can you enable me to share my screen? It won't let me share my screen.
It won't?
No, it says host disabled screen sharing.
Okay.
Yeah, that's the default anymore for the meetings because of people who are going.
Okay. Who can share? I only have a choice of all participants or only hosts, so hopefully nobody else shares.
Yeah, hopefully not.
Who can share and someone else is sharing? Only the host, so that way only I can cut you off.
Okay.
You could attempt to add her as a co-host of it.
I tried that because she's got a student email address, it wouldn't let me.
Oh,
I don't know why.
This works.
The wonders of technology. You can do this, but
yeah, no, I think this will, you should be able to share now. And then we can probably pause the recording.
Of deep underground biofilms at the Sanford Underground Research Facility. Take it away, Bethany.
Okay, thank you, Shane. Okay, I'm gonna hide everybody's video. I want to preface my talk today by saying that I'm hugely disappointed that I don't get to see you all in person. I had fantasized about this day for a really long time, so I don't blame anyone in particular, just the whole entire world. So since we're living in unconventional times, I would like to start off in an unconventional manner with a song. Oh, if my thing's gonna work. There we go. Hold on, having technical difficulties.
Should I play the guitar?
Yeah.
Maybe.
I guess we haven't worked out the chord progressions though, so maybe.
Wouldn't know the difference, Shane.
That's true.
Okay. I'm not able to progress through. Oh, there we go. With a song. You get a slide, I get a scope, Cindy. You get a slide, I get a scope, Dave. You get a slide, I get a scope. We'll go picking for a small nematode, Cindy. Dave, oh my. Written by me, inspired by this project, and dedicated to my committee. Custom amplicon sequencing of deep underground biofilms at SURF. Now, here's me. Yes, that's me talking today. And this project was the brainchild of Oksana. And with much help from Dr. Anderson and then my advisor, Dr. Sarver, and of course not listed Dr. Bergman, he also accompanied me on many forays underground. And I was looking at deep underground biofilms, 1 mile underground, and looking for what was living inside these biofilms and what made up these biofilms. And this took place at Sanford Underground Research Facility in Leeds, South Dakota. And the reasons we want to look in strange places like deep underground, well, for one, this biome, if you will, this deep underground biome has only been studied quite recently. And by that, I mean since the 80s. So it might not seem recent, but in the whole history of science, it is relatively recent. And it's a way to explore our biosphere and extend that definition of biosphere. And then any organisms, bacteria or other organisms, if they're novel, could give rise for medical research or industrial research or other molecular research. And then also the deep underground can serve as an analog for astrobiological forays, or maybe looking at Mars, looking underground at Mars, so it can serve as an analog for that. And I did this by DNA sequencing and microscopy. So I'd like to give you a little bit of background on the Homestake Gold Mine. So this was the previous life of Sanford Underground Research Facility and how it went from Doozle to Soozle to finally surf. So about 140 years in brief, the Homestake Gold Mine is in Leeds, South Dakota, which is in the northern Black Hills. It was in operation for about 126 years, and it was the largest gold mine at the time in North America and also in the Western Hemisphere, and then also the oldest gold mine as well. The deepest shaft goes down to about 8,000 feet, and there's about 370 miles of tunnels that stretch out from the mine, and over the course of its life, there is well over 40 million ounces of gold and actually 9 million ounces of silver that came out of that mine. But the mine closed in 2002, and it closed because of low gold prices and poor quality ore. And I believe at that time, it was the late 90s when they announced the closure, gold was like $280 an ounce or something. But in its new life as a deep underground science facility, there's currently a bunch of science going on. However, in the 1960s, when that mine was in operation, Dr. Reman Davis was doing solar neutrino experiments on the 4850 level. And so now we flash forward into the 21st century, and we have Sanford Underground Research Facility is now operating where that Homestake gold mine used to be. And the site was pegged for the science facility since the mine's closure, but since they, the ball didn't get rolling right away, they stopped dewatering the mine. And what that means is they turned off the water pumps. So if you have a giant hole in the ground that's below the water table, it's going to fill with water. And so the mine, it was really expensive to keep those pumps on, so they turned them off, and the mine ended up flooding. And it flooded, I believe, to the level 4,600 feet below the earth, so it flooded quite a bit. But then in 2006, the property was donated by Barrick Gold Mine, which is the parent company of Homestake at the time. And then the state of South Dakota and T. Denny Sanford donated a bunch of money, and then the South Dakota Science and Technology Authority was created. And that is sort of a quasi-government body that oversees the operation of the mine. And then in 2007, the National Science Foundation made the Deep Underground Science and Engineering Lab, which eventually became Sanford Underground Research Facility. And this is currently where a bunch of physics experiments are going on, world-renowned ones with a lot of participants about dark matter and solar neutrinos and a bunch of fancy physics stuff. I can't get into that. If you're interested, there's a wonderful visitor center up in Leeds. So how did this project came about? I came onto this project, well, it started in 2016. And it started because I was whining and complaining to Oksana that I really wanted to go underground. And if I can get sentimental for just a second, I graduated from high school in Leeds in 2001. I know it doesn't look like it, but I did. And I remember the day that they announced the mine was closing. I believe I was a junior in high school, and it was absolutely devastating. It was a tragedy for everyone that lived in that town. And so I wanted to be able to go down there and see this mine that had always been up there on the hill and had been a part of everyday life. So I convinced Oksana to take me underground, and she said, hey, look, let's do a survey of the mine, the old tunnels. And so we surveyed 5 different levels, and we were looking for pretty much anything that looked like it was alive. And in our case, we wanted to look for eukaryotes to contrast to prokaryotes. So prokaryotes are organisms that are bacteria and archaea. So single-celled organisms that don't have a nucleus. So contrast that to eukaryotes, which are everything else, protists, single-celled eukaryotes like protists, animals, plants, fungi, humans, we all have eukaryotic cells, and it just means we have a nucleus, that we house our linear DNA inside, and that we also have membrane-bound organelles such as mitochondria. So we were looking for these higher taxa, and we ended up finding a few things, so some fluffy things and some squishy things and some slimy things, and we collected them, brought them back to the lab, and analyzed them, did some culturing, did some microscopy. And so that's where my project all started. So then I decided for this particular project, for my thesis, that I would focus on one level, the 4850 level, so 4,850 feet below the surface, not quite a mile, on a particular site called the 17 ledge. Now, I'm going to define what a ledge is, because when I first heard that term, I was picturing like being underground and then there being like a giant drop off, like a cliff, which is absolutely ridiculous when you're in a underground mine, but that's what my mind saw. So I just wanted to define what a ledge is for you. Now, any geologists in the house, please just bear with me because I've only taken one tiny geology class, but here is the beautiful Black Hills in Lead, South Dakota, is up here at the northern part. And then the larger geological feature is called the Lead Dome, and that is where the Homestake mine sits. And this is a longitudinal section of that Lead Dome, and then this gray squished looking stuff is, let me get my laser pointer here. This is the Homestake Formation. So this is where parts of the mine actually are dug into this rock. And because I love geological terms so much, I would just like to read this to you. This is the type of rock that makes up the Homestake Formation. In upper green schist facies, siderite phyllite is dominant. In lower amphibolite facies, grunerite schist is dominant. I love words for rocks. I think they're beautiful. But anyways, that's not important. I just wanted to share that with you. So in this map here, this is the surface up here. We have our Homestake Formation rock, and then here's the 4850 level, this line. So I just want to amplify that for you. And then this little section here, this little piece of rock, if we follow it up with our eyes, we see it's called the 17 ledge. So a ledge is just a fold in that type of rock. So it's just how the rock is folded, and they call it a ledge. And so that ledge intersects with the 4850 level, and that is the particular area that I'm sampling from. So here is an overall view of the Homestake Gold Mine. We have the Yates shaft here. So this is that big silver tin can you see on top of the hill, and that is where you would find this cage, also known as an elevator. We'll call it that. Cage kind of sounds scary, but you get in that cage and you go underground. It takes about 15 minutes to go from the top to the 4850 level. It's a little bit noisy. Sometimes there's water dripping on your head. And once you get to the bottom here at the 4850, you get off, and then we hop on a locomotive. This is just a little choo-choo train. There's an engine connected to the other end, and then you can go flying down the train tracks. You could also walk, but it's when you're underground and you have like no context for where you are, it's kind of gets a little disorienting, and it can feel like you're walking forever. So thankfully, the surf staff provides us with a little locomotive. This is a picture of the 4850 level, so just kind of a bird's eye view. So here's the Yates shaft where you get off your cage, off the elevator, hop on the train, and then you come down here through this tunnel and you pass by the Ross shaft, which is where the Ross campus is, and the Black Hills Underground campus is there. So there's this nice little clean room there. We can go do microscopy. Right next door, you would find Brianna Mount doing her low background counting. But this is our little lab that we can work in when we're down there. And this is just a view from looking in, looking out at the door. So that's a nice little facility. We swing by there, and then we come about another mile away from all the civilized areas here. This area up here is like, it looks like you're in a building. There's lights, there's walls, there's a flushable toilet, there's all sorts of stuff. So it doesn't really seem like you're underground until you get way out here. And then this tunnel here is where I'm sampling from. This is the 17 ledge, and this is just a little map of that tunnel. The train pulls up right here, and then you can very, very nicely unload your gear on this table because it's very muddy and wet out there. And then just trot down the cave here. And then, so this is where I took some of my samples from these little cutouts in the side. That's what these little half circles are. And they're just little areas where miners in days bygone, they would just make these little cutouts in the side of the tunnel and then drill boreholes in them. Oh, and then once you get to the end, this is what you see. There's some lovely, beautiful biofilms growing on the walls of the rock. So this particular area, the 17 ledge, it is known as a red zone. My guide Tom would say, well, it's more of an orange zone, but red zone sounds scarier, right? But nobody's really allowed to go out there except for the maintenance guys and then us crazy scientists that want to go out there. And that's about all that the people that go out there. So it's not a high traffic area. It is very hot. It is very humid. Now, if anybody's from the South or has lived in the South, you're probably thinking, yeah, that's like a nice spring day, 91 degrees. But for me, who is temperature sensitive anyways, it's gross. I don't like being out there for very long. And then once you walk through the tunnel, there's like a temperature and a humidity gradient. So it gets hotter and more humid the further you go into it. And about 20 minutes is the max that a person can stay out there. It's wet and muddy. There is ankle deep mud and even deeper water. You could lose a boot or twist an ankle. It gets pretty hairy out there, and it's stinky. It smells like hydrogen sulfide gas. It's absolutely miserable. If you don't believe me, you can just ask E. He was just as miserable as I was. I do have permission to use this photograph from him. This is for a class. We went down there with Dr. Bergman and Reman for a class and took all the grad students, and E was not having it. He was very uncomfortable. So anyways, these biofilms grow on the rock walls, and they grow from this water that leaches out, and the water comes out from either boreholes or just cracks in the rock, and then where this water leaches out, these biofilms grow. And they are just as slimy as they look, kind of really snotty and boogery. Okay, so what are biofilms? I keep saying this word, and I'm sure most of you have heard it and you know what it is. But if you just do a quick Google will call it a thin, slimy film of bacteria that adheres to a surface. Yes, that is what it is. But it's so much more than that. They're so very complicated, and I will not go into the architecture of biofilms, but I will just say they are like a little city of microbes all living together and working with each other in concert. You know, probably there's some that don't get along, but for the most part, it's this little functional community. And you'll find biofilms in many places. You'll find them in bodies of water, growing on rock. You'll find them in the bottoms of rivers and lakes, in your home, in your drain. There's biofilms in your drain growing. There's biofilms in your shower head. If you don't want to know any more about that kind of stuff, I would not recommend reading the book Never Home Alone by Dr. Rob Dunn. It'll creep you out. In your oral cavities, the plaque on your teeth, that is biofilm. Also, we find biofilms in hospital settings where we absolutely do not want them because they cause disease and sometimes death. They will grow in catheters, in ports, in medical equipment where we don't want them. And also, we use them on purpose in industrial settings. So like in wastewater treatment plants, we purposely cultivate bacteria in biofilms to help us clean the water. So as briefly as possible, because methods are never that interesting, we collected 6 biofilms from the 17 lenge, put them in sterile conical tubes, took them back to the lab, did microscopy, DNA extraction. And then I didn't mention this before, but we collected surface samples. And the reason we did that was because there has been some speculation from hydrogeologists here and there that possibly the surface water from like water features such as Whitewood Creek and other creeks in the area could leach into the groundwater. And so we collected 6 surface samples from Whitewood Creek along Kirk Road, and we collected sediment, rock biofilm, and we filtered some creek water. And in case you don't know where Kirk Road is, okay, this is Leeds, South Dakota. Here's the open cut gold mine. Here's Sanford. Here's Chubby Chipmunk. You guys know where that's right. So if you pass Chubby Chipmunk and go down 385 south, like you're going to go to Custer, but then there's this little road here. This is Kirk Road, and it follows Kirk Canyon along the backside of Lead. And when I was in high school, we did high schooly things when we drove down that road. But if you drive all the way down, you come out here on 85 south, you can go to Louis and have a burger and a beer if you're familiar with the geography of Lead. But anyways, so we sampled Whitewood Creek, which follows that Kirk Road, in 2 different spots. And we sampled, I chose Whitewood Creek particularly because it crosses over that surf property. And so I told you we sequenced DNA. Well, we sequenced part of the 18S ribosomal RNA gene. And this gene's about 1,800 base pairs long, give or take, depending on the species you're working with. But I chose this gene for lots of reasons. There's lots of information on it already, but it's a highly conserved gene. But we couldn't sequence the whole thing because our sequencing platform that we used is limited to about 500 or 600 base pairs or so. So had to choose some shorter regions. So these V regions that you see here are variable regions, and there's 9 of them in this 18S. So the 18S, excuse me, I didn't mention, is that small subunit for of ribosomal RNA for eukaryotes only, the 18S is. And so I chose 2 variable region 4 and 9, through a literature search and then also some primer testing. The V4 region is the longest region, and so that is why it worked well for this project, has been known to amplify, not quite universally, but many different organisms. And then the V9 too is recommended for amplifying protists. So in addition to those 2 sequences, we sequenced this ITS region. Now this is an internal transcribed spacer region. This is a gene that's used to verify fungal taxonomy. So that's why we chose that. If there's any fungi in our biofilm samples, we wanted to be able to see what those were. And this is just a gene that's housed between the 18S gene, which is the small subunit ribosomal RNA, and then the 28S, which is the large subunit, and then the 5.8S, which is just another subunit of, I believe, the large. Dr. Anderson, don't get mad at me if that's wrong. And then also, in addition, we sequenced the 16S variable regions 3 and 4, and that is the paralog of the 18S, but it's for bacteria. So we wanted to see bacteria, eukaryotes, as well as fungi too, what were in our samples through DNA sequencing. Now, library preparation. So this is how we prepare our DNA samples for sequencing. I want to go over this, not because I'm a salesperson for Illumina, but because we have this machine in our CCBR lab in the life sciences building. And I feel like when you hear the word, you know, next generation sequencing, mass parallel sequencing, it sounds like, wow, that's really high tech and complicated. It is high tech, but it's not complicated. And any student that has a modicum of benchtop experience and a moderate amount of supervision can do this. And what you do is you take your sample. In my case, I had a biofilm sample. I extracted DNA. And then within that DNA elution, there's maybe thousands of species of their DNA is in that little tube. And so then you amplify a gene. So in my case, the 18S genes and 16S and the ITS through polymerase chain reaction, PCR, the absolute cornerstone of molecular biology. And this is just a way to take a chunk of DNA that you're interested in and make billions of copies of it. And that's, you want billions of copies, so you have a lot of DNA to work with. And then in this PCR procedure, what is actually done is to that PCR mix, little tags are added. And these are just very short nucleotide sequences that tag each sample. And that way, when they're sequenced, they can be sorted out later in data analysis. So each sample is tagged with its own special little barcode. And then that way you can pool all of your samples together, put them all in the same tube. It sounds crazy because when you're working with DNA, you're like, you don't want to cross-contaminate, but that is what you do. So you can pool all your samples together because they're all labeled individually. And then, bing, bang, boom, you put them on the MSIG. So this is the Illumina MSIG. There's a little drawer right here where the sequencing chip goes. This is just a little glass chip, and that's where the magic happens. Just kidding, it's not magic, it's science. And then there's a little door here, and that's, you open that, you put your sample in there, and beep, bop, boop, you program your computer, and next thing you know, 24 hours later, you get data. And this is what your data looks like. It's a FASTQ file. I don't know what the FAST stands for, but Q stands for quality. And it's just a DNA sequence, and then its name, so it's labeling it for you. And then all of this gobbledygook down here is quality data about each nucleotide. So that way, when you feed it into your analysis software, it can determine if your sequences pass a quality, if you do like a quality control. So data analysis on the CLC Bio workbench. Data gets imported onto the computer. This is another wonderful resource we have in the CCBR. Quality control, so you take a look at your data. Is it good? Does it pass quality control? If it doesn't, you can trim it and get rid of it. And then merging, I'm not going to talk about that. It's not important right now. However, I do want to define OTU clustering and what an OTU is, because I'm going to be saying that word a bunch, and just like to clarify what that is. Okay, so OTU stands for Operational Taxonomic Unit, and it can be thought of as a hypothesis for a species, but in my case, I'm going to generalize it even more and say it's a hypothesis for a taxonomic group. So an OTU is just a way to take your data. So for instance, these are all of our sequences that we get when we're done sequencing in our data file. And it's a way to sort them by some sort of likeness. Well, in this case, I've sorted them by 100% likeness. So the sequences are similar, but they're not 100% the same. So to make an OTU, you tell your computer software, okay, please sort these by 100% likeness or whatever likeness you want your sequences to be. And then what it does is it takes a consensus or an agreement of all those sequences, and it just gives you one sequence as an output, and that's just called OTU1, OTU2, or whatever name is given at that point. So it's just a way to pare down the data, because after you sequence, the output you get is hundreds of thousands, if not millions, of DNA sequences. That is a lot of sequences to work with. So you'd want to be able to pare that down, because you don't want to have to individually analyze each sequence. But not only that, there are going to be duplicate sequences within your data. So you just want a way to sort them and pare them down so it makes it easier to analyze. Oh, I thought I'd never get here. Okay. There are older and fouler things than orcs in the deep places of the world. If you don't know this quote, you need to, after I'm done here, go watch all 10 hours of Lord of the Rings. Anyways, Gandalf was referring to the Balrog. However, in our own deep places of the world here in the Black Hills, we saw mini creatures. And so in this case, I'm going to show you some microscopy from the underground biofilms only. So this isn't surface samples. This is just what I saw in the underground biofilm. So organisms like nematodes. So nematodes are pretty much ubiquitous. You're going to find them anywhere and everywhere. They can be pathogens and parasites of plants and animals and humans. If you're bored, do not Google human nematode parasites, because you'll have nightmares forever. It's very diverse and disgusting. Anyways, nematodes are found everywhere. In this case, they were found 1 mile underground in these biofilms. Rotifers. These are just my favorite microscopic invertebrate. I love rotifers so much, and I can't even define why. I just think they're adorable. These are also freshwater species. They would be found in rivers or lakes or ponds, anywhere there's water. Flatworms. So this is a large phylum from the platyhelminths. These flatworms, they're related to things like planaria. So anybody that's taken a Gen Bio class, you'll have worked with planaria. Those were a flatworm. This guy is also a flatworm. When we first saw it, we didn't know what it was, and we called it an alligator because it kind of looks like an alligator. It's got that same shape, but it's not an alligator. It's a flatworm. These are also cosmopolitan freshwater species. In this case, found 1 mile underground in some water. And then gastrotrix. These are also freshwater species, and this guy I like to think of as a hairy pickle with a stinger because he looks like a gherkin had a baby with a bumblebee. He's so cute. Adorable. I love tiny things. If you ask my mom, she would say, Bethy loves tiny things. And it's true, I do. I love the microscope. So moving on to single-celled organisms, we also saw many protists. These are single-celled eukaryotes. This is a flagellated protist. You can see its little flagellum here. It's probably sensing its environment or searching for prey. And then also we saw testate amoeba. So these are also single-celled eukaryotes, a protist. And they kind of look like olives. For some reason, every time I see these, I think of a martini with like a skewer with these amoeba on them. And so these are just an amoeboid cell, and what they do is sometimes they'll either secrete like a calcium carbonate test or shell, or it can be silicate based, or sometimes they agglutinate it from their environment. So they'll pick up little particulates and glue them together and make a happy little home for themselves. And you can see them also here in this scanning electron micrograph as well. see the little aperture there. There's one there. There's one right there. Ciliated protists, so once again, a single-celled eukaryote. Unfortunately, Ciliophora, or I believe they're a phylum, and I can't get any more specific than that just by looking at it under the microscope, but I know it's, excuse me, a ciliate because it has all these cilia. Okay, so comparing that microscopic data to our next generation sequencing data, what I'll present to you now is just for that V4 region of the 18S. And the way I clustered my OTUs was by 97% similarity. That 3% error it can, for a couple of different reasons, there can be some variation in that 18S gene, even within a species, but also because it accounts for sequencing error too, although the platform we're using should be high fidelity and we shouldn't have too much sequencing error that we can't filter out. And then, so once the OTUs were clustered and made, I bounced them off the Silva database. So this is a database of the 18S and 28S genes. It has hundreds of thousands, maybe even the newest version has millions of sequences now. And so what I said is, well, okay, well, if my OTU consensus matches the database at 97% or greater, then I would like information about that, please, and thank you. And so for the total sequences, this is just a lovely sunburst chart here. And the total sequences that were put into OTUs, 24% of those sequences could not be identified at some sort of taxonomy at 97% or greater. Now that could be because the database doesn't include every organism on Earth yet because there are millions of different species, or it could just be, I don't know, there could be several reasons, but anyway, sorry. So we see that a small portion of our sequences go to these three groups, which are protist groups, Amoebozoa, Centrohelida, and SAR, which is, these are like sub-phyla. They're not, or excuse me, sub-kingdom. They're not phyla. Protists are like this enormous junk drawer of taxa. There's a lot going on in there, but they are all single-celled eukaryotes. And then this orange is Opisthoconta. So Opisthoconta is a clade that includes animals and fungi, does not include plants. And so if we get rid of those unclassified sequences within our sequence data, so along this outermost circle here is identified to class. And this purple is rotifers, the red is gastrotrix, the yellow and the green are two different classes of nematodes, and this blue is flatworms. So what we saw under the microscope for the metazoans being multicellular, which are true animals, that's what metazoa are. And so anything that's multicellular that isn't a plant or a fungi. And so all those larger organisms we saw under the microscope, we also saw in our sequencing data. So our sequencing data isn't saying this is what this is, it's just saying your sequence matches this sequence. And so there's a possibility that that's what this is. So then if we look at a bar chart, so this is just the underground biofilms for the metazoa only, so for those multicellular animals only. So we also see we do have a portion attributed to each sample here that is unclassified, not able to be classified for whatever reason. The purple is our rotifers. So we see those in all the samples. It's in this one too, it's just very tiny bit. I want to point out that this says abundance. Now, this is just the abundance of sequences. It doesn't correlate to the abundance of organisms in a sample. So it's kind of deceiving when you look at it. You might think, oh, there's more rotifers in this sample than the rest, but that just means there's more sequences attributed to the rotifers, and that happens because that 18S gene has a variable copy number. So there can be, some organisms can have 1 or 2 copies of it in their genomes, and some can have thousands. So you can see when you go to amplify that DNA, how it could preferentially amplify something that is more abundant in the genome rather than the organism itself. We had matches to 5 different orders of nematodes. A couple of them were distinguished to genus. So this one, this rhabditid nematode, and then this one as well. These other 2 are ambiguous. That just means that it could only be classified to this order. So that for this nematode, the Areolamida, this one, it could only classify it to the Rhabditida, and then this one, the Treponcida. We actually saw two different matches to two different genera of flatworms. So this Catanula flatworm and the Stenostomum. It looks like in this sample that sample 400 here is the only sample where we saw that Stenostomum match. And then our Gastroctrix, we saw those in all of our samples as well. So comparing the surface samples that we collected from Whitewood Creek to the underground samples, just to see if there was any overlap in taxa. Now, there, yes and no. So we saw nematodes, of course, because nematodes are everywhere. But in the underground samples, it was specifically rhabditid nematodes. So that's a order of nematodes. And then in the surface samples, strictly monohysterid nematodes. So there was no overlap in taxa at a finer resolution. Flatworms, so underground, we saw strictly Catanula, that Catanula species matched to the underground samples. That was not seen in the above ground samples. They just had that match to the Stenostomum genus, and then there was some other flatworms in the above ground samples as well. The nematodes from the class, sorry, Inoplia, they were mostly seen underground. There were a few reeds in an OTU from the above ground samples as well. Gastroctrix, we saw that same Chitonitis genus in both the above and below ground samples, and the same for the rotifers in this Ploimeda group of rotifers. And then a bunch of protists. Actually, there wasn't a whole lot of overlap in the protists above and below ground, but above ground we also saw, you know, it was a creek. We saw crustaceans and insects, oligochaetes, which is a phylum that includes leeches and earthworms and other worms, arachnids, there were mites in there, and then lots and lots of OTU matches to protists in the above ground samples. So the above ground samples were more diverse, and that was expected, of course. So just a little bit of microscopy from the surface samples, because microscopy is fun. Nematodes, of course, were seen in the surface samples. And some sort of insect larvae, probably a fly or a midge. Dr. Downing's looking at this, and she could probably tell you better, but just from my brief entomological knowledge, looks like some sort of fly or midge larva. And then diatoms, these are just so pretty. I love them. These are an algae, so they're single-celled, they're a protist, and I just think they're very beautiful. You'll find them pretty much in any fresh water. So if you're bored today, go collect some pond water and do some microscopy. Now, alpha diversity, so this is just for the underground samples, and this is a measure to say, did we sequence enough to see the full diversity in our samples? And the answer is yes, probably we did. And we know that because this, the graph, the line kind of drops off. If it continued to go up, we would think we probably need to sequence more, but more than likely we saw the full diversity in these samples. This measure can be skewed by, of course, how you analyze the data. And in this case, I cut off any OTUs that had fewer than 10 sequences in them. Sometimes by doing that, it can skew your alpha diversity. However, sometimes if there's just two reads, like 2 reads out of 1 million that are identical to each other and nothing else, they could be sequencing errors and not necessarily rare sequences, or they could be some sort of other error that didn't get filtered out. So I just chose to cut those off. Now, a brief mention about the prokaryote. So about the bacteria that we saw in the samples. So this is just for the underground samples too. I actually did not sequence 16S for the above ground samples. There was 97% OTU similarity with the bacteria, and then I chose to do an 80% taxonomy match just to give us a better snapshot at the phyla level. So just some notable phyla from the 16S data, proteobacteria. So these are a very diverse group of gram-negative bacteria. You find them in many different types of samples. Some of them are obligate or facultative anaerobes, and then a lot of them can be chemoautotrophic or chemolithoautotrophic, just meaning that they use inorganic electron donors. 2 of the most prevalent OTUs from this proteobacteria phylum matched to Thiothrix, which is a genus of bacteria that they oxidize sulfur, and then also a match to Methylococcus, which is a genus that is methanotroph, which just means it uses methane as its main source of energy. These bacteroides, sorry, my Latin's rusty. These are found in soil and water habitats. They are known to degrade high molecular weight organic compounds such as polysaccharides, proteins. The nitrospirene, these are nitrifiers, and some can be sulfate reducers and some can be magnetotactic, which means they can take a magnetic element such as iron or magnesium and sequester that in their little bacterial cells. And so the nitrifiers are known to oxify ammonia to nitrate and then eventually nitrite and then eventually nitrate. Oh, this one's fun. Chlamydia, yeah, that's because the genus Chlamydia that causes the human STD is in this phylum, but this phylum, from what I understand, they're all obligate intracellular parasites of eukaryotes, so that's fun and exciting. However, I have seen this phylum pop up in other underground research, even at SURF too, so that was interesting. And then the last one that I thought was interesting is this Verrucomicrobia. This is a phylum, I believe it's a relatively new phylum, but there was some research about 10 or 15 years ago where they discovered this Candidatus nematobacter. And this was an OTU match that I saw in this phylum. And so this is a known cytoplasmic endosymbiont of parthenogenic nematodes, very specific. So parthenogenic just means there doesn't need to be sex to be babies. So no sperm is involved. A female can lay an egg and it can magically turn into a baby. Just kidding. Once again, not magic. And this parasite, or this Verrucomicrobium, is inherited specifically through the egg cells. So those are just some notable phyla that I saw in my underground samples. And then I compared this, I don't have any charts or anything, but I compared it to some other underground research done at SURF, and very similar phyla pop up. Okay, the best part, you guys. This is the most exciting. So when I started this project, I didn't do it because I thought I wanted to find like something new and cool and crazy, but I read some literature from Gaëtan Borgoni. I believe he is in Belgium. And in 2011, he published this paper. He had gone down into some South African gold mines, which are very deep, and he went about 1.3 kilometers deep and he filtered some borehole water, and he said, hey, you guys, I found a new species. It's called Halocephalus mephisto. It lives underground. It's native to the underground. It's been there a long time. And from what I could tell, he had filtered that borehole water. He found one worm. He took it back to his lab. He cultured it in vitro, and supposedly he could do that because it was a parthenogenic species. And then he sequenced, which I'm assuming is the culture. And he said, boom, I got a new species, Halocephalus mephisto. It's called the devil worm. So if you Google Halocephalus mephisto, you'll just see publication after science publication about this guy and the deepest living animal, H. mephisto. So you've already seen this. I showed it to you right here. So I had one of my OTUs in one of my samples, sample 400, gave me a 98 to 99% match to Borgoni's H. mephisto. So what I did is I reclustered my OTUs at 100% so I could see how many unique sequences matched this GenBank match to H. mephisto. And I got 4 OTUs that matched anywhere from 98 to 99%. And you can see right here in the middle of the sequence, It's where the majority of our differences are. There's a couple from about base pair 110 right here to about 130. There's a couple of insertions and there's a few SNPs. And this isn't quite the middle of the sequence, so I'm not sure why these are all here, if they could be sequencing errors or if there's some other explanation for it. But so, and my sequence matched his better than it matched anything else. I actually didn't get any other matches. So this maximum likelihood phylogeny shows my 4 OTUs here nestle quite happily on this Panagrolamidae nematode family tree with Borgoni's H. mephisto. And then the next most recent relative are from the same Halocephalus genus. However, this one here is, I believe this one's a termite endosymbiont and this one's an equine parasite. So those are the two most closely related in the same genus. And then the rest of sequences from the same family but different genera nest out here on our neighbor joining tree. Okay, so here we are. We started at 1 mile above sea level in Leeds, South Dakota. We went about a mile underground, collected biofilms. We're miserable and muddy and dirty, but when we looked under the microscope and through our sequencing data, we saw these eukaryotes, these single-celled protists, and all sorts of adorable and wonderful micro eukaryotes. So now what? So with that established, future research could look into the water source or the origin of the water to maybe make some ideas about where these organisms came from, if they came from the top and went through the rock, or if they came from anthropogenic sources. Looking at the water chemistry, full disclosure, I do have water chemistry data. I was not able to use it with my sequencing data, but it was collected. So additional water chemistry information to go along with sequencing information. Additional surface studies, so maybe looking at other areas on the surface and surveying what types of organisms live in the other surface environments. And then also looking at other areas of the mine. So there were other biofilms on different levels of the mine that Oksana and I initially saw too. So actually doing some deeper digging into some of those other areas. No pun intended. And so some other genomic ways we can analyze these biofilms. So a high-C metagenome. So this is just a way to get metagenomic data. It's a little bit fancier than what we have on our campus. However, we have previously sequenced a sample using this method at a different lab, and it worked nicely. Genomic mining for novel taxa. So this is just a metatranscriptomic approach to comb through the data and look for maybe any novel species through that method. Also culture-based studies looking for antibiotic production could go on from here. And then my favorite, and I would love to do this, whole genome sequencing, especially of that nematode that matched the H. mephisto. And actually H. mephisto's genome was sequenced recently and published last November. So that information exists, and I actually found it on GenBank, so it's there. And so that would be fun to compare my nematodes to that whole genome sequence of H. mephisto to maybe try to see if we do have the same species. And if so, that would be very exciting because mine's deeper than his. Okay, that is it. We made it through. I hope I didn't go over my time. I think just about perfect. Thank you so much to everyone, especially Surf, for letting me go down there and trudge through the dark and the mud. Thank you to EBSCOR. Because of EBSCOR, well, I mean, they funded my project, but also I was able to go to the Biology of Genomes conference in Cold Spring Harbor in New York. Absolutely fantastic, amazing experience. If anybody wants to know about it, we can talk later. And REU students, I had a whole troop of them over the years. Thank you to them for participating and helping. Oksana, your brain, thank you for that. And Dr. Gable and Dr. Lamb, thank you for letting me use your microscopy equipment and helping me with it. And to my MSIG compatriots, you guys didn't really do anything, but you were there for moral support and comic relief, and I love each and every one of you. Okay, that is it, and I guess I'll get out of here and we will take some questions if anybody has questions.
yeah, if you want, you can either just speak up, or you could also put the questions in the chat that Bethany can see.
Super
Okay.
job. Super, super job, Beth. I'm going
Thank
to
you.
pause. Woohoo! I do have one quick question for you.
Okay.
So you talked about the reason that you sampled surface water and underground water was because some people hypothesize that the surface water may trickle down. Do you know from any other published or unpublished studies what the age of the water at some of those deeper levels at the 4850 actually aged to?
Yeah, from that particular area, there has been some studies, and I want to say 10,000 years old. However, there has been some models in hydrogeology that say the surface water does mix with that old water, but at a very slow rate, so probably decades. You know, that meteoric water does eventually percolate down and mixes with that old water. But yeah, the older storage water down there is about 10,000 years old.
So I have a question about the communities. So you were kind of putting your samples together, but in the different biofilms, that some were fuzzy and some were slimy and some were this and that, can you actually separate out who was where, and so which species were interacting in the same community or
Yeah,
living
like
in the same?
the higher taxa, is that what you mean?
All of them, just everything that would be in a particular, can you separate it out by basically ecosystem of where these different organisms are?
Possibly, and I thought about doing something like that, like even making some sort of map to map along that tunnel, you know, in which biofilm, what organisms reside, because there are some differences, and I didn't show that, but there are some differences in the types of organisms that live there. Like there was one biofilm that had specific matches to specific taxa that didn't show up in any other biofilms.
Because it would be interesting to kind of learn more about those communities and how the individual species are all interacting with one another.
Yeah, and compared to like the bacteria as well, like the composition of the bacteria versus the eukaryotes too. Yeah, that would be.
Looks like Yoon has a question in the chat, Bethany.
Okay. Hi, Yoon. Okay, Yoon says, I was wondering which organism is responsible for making the biofilm. So like the slimy part that you see is bacteria and many different types of bacteria, but it's also EPS, which is extracellular polymeric substance, and it's secretions from the bacteria that are polysaccharides. There's proteins in there, there's exogenous and DNA from critters that die inside there as well. So like the greater matrix of the biofilm is bacteria, but as far as like which specific one started it or is responsible for the majority of it, I'm not sure, but yeah.
That is sort of, yeah, you bring up a good point. It would be nice to know chemically what the EPS is, and if anybody is feeding just directly on the EPS, it's maybe more than just a matrix that things are moving about in or attached to.
Yeah, biofilms are incredibly complicated. Any other questions? A chironomid larvae. Okay, thanks, Dr. Bergman.
I have a question, Bethany. Of course, we've talked about a lot of this stuff, but for those, you know, that when they discovered that nematode in the South African mine, that was a huge deal. There was a whole series of papers published in Science and Nature. It was picked up by National Geographic, the BBC, and the possibility that we have the same thing here is a pretty big deal. You know, it raises some really big picture questions, you know. What would be kind of how would we nail that down?
We would probably have to have some sort of morphological identification, which you and I can't do because we're not nematologists. In order to identify a nematode, you have to like look at its vulva and its mouth and all sorts of stuff. Very complicated. So we'd have to have somebody identify our nematodes for us, and then I would love to do a whole genome sequencing on it and compare it to the one that was done in South Africa.
I have a question. Go ahead. Sorry, Mike.
Can you tell how rare that particular nematode is compared to the other ones?
Well, it supposedly hasn't been seen anywhere else on Earth.
No, just within your sample itself.
Oh, yeah, it is pretty rare. So as far as the sequences that are attributed to that match, there weren't very many. So if I just had to guess, I would say it's probably not a very prevalent nematode in those samples.
So you have a tough time looking through all the nematodes and saying this is the one.
Yeah, so they are big enough that you can look at them under a stereo microscope and pick them out with a micropipette. And so then you could work with individual nematodes, but it would be very tedious because you'd probably have to pick out, a few hundred of them to do that.
Beth, I have a question. I've read that worms, flatworms, can live in ice and frigid conditions by making higher levels of proteins that they secrete into their hemolymph so they can survive, not freeze. But what about at these hot and humid depths? Do you see anything on how do they adapt to live in those kind of conditions?
Oh, yeah, good question. So I don't know. The water itself wasn't particularly hot, the air was, but so not a flatworm, but the nematode in South Africa, that's why they sequenced that genome, because they wanted to look at these HSP70 proteins, these heat shock proteins. And supposedly that particular nematode produced a lot more of those heat shock proteins. So I imagine if there's other individuals like flatworms, they might have that same, those same genetic components, those additional heat shock proteins. If they're truly adapted to that sort of extreme environment, yeah.
Liz, you had a question?
Oh, I did, thanks. I was wondering, in the part of your data that you showed, there was a whole bunch where maybe there wasn't anything in the gene bank that matched what you had. You couldn't really tell. Is there any value to checking your samples again, say in 5 years, to see if more organisms have been added to that gene bank?
Oh, for sure. Just comparing, so like the nematode sequences I pulled off GenBank to make my tree. Well, I did that because Dr. Borgoni had done that when he discovered his H. mephisto. And I noticed that I had way more sequences than he had in his tree. And that's because that 18S gene, I don't think 10 years ago there was as many sequences available. So definitely, yeah, as time goes on, there's more sequences uploaded.
That was actually the most frustrating part of this project. The groups of organisms that we found were had the poorest representation in GenBank.
Yeah.
And so that turned out to be, we thought we'd be able to do a lot better in terms of the identification of the things we found down there than we were able to do.
Mm-hmm. Oh, along those lines, Beth, did you just blast some of those not assigned sequences against GenBank?
Yeah, and so I did get some matches at like a lower level, like a 95% or 90% match to some of those. But some of them I didn't get any matches at all, and I wonder if maybe because some of those sequences weren't filtered properly, like the OTU clustering is supposed to filter out chimeric sequences, but part of me wonders if maybe that didn't happen. I'm not sure. Yeah.
Well, if they're really abundant, perhaps this might be valid ones.
Yeah.
Hey, Beth, I have a question.
Justin?
Yes.
You know what? I had a gel picture in here just for you, but I had to take it out because I know you love them.
Oh, I love gels. Yes. Reminds me of Jello. Question for you. So when you have, you recover some number of sequences, and you were right to say this doesn't scale, exactly with the number of organisms in the sample, but it's probably not unrelated either. Has there ever been an attempt to like either simulate a community based on the number of, ITS, ribosomal genes that are in the different genomes, or empirically like make a community and then sample it with next-gen approaches to see how well the number of sequences recovered actually matches the actual composition of the community? Because it seems like there's got to be somewhere between it doesn't mean anything and that you can scale it and interpret it at face value.
Yeah, so there has been many publications with sequencing mock communities and even single cell sequencing, and it has been showed that even within like a single genome, multiple OTUs will be identified, and that's it probably just because of SNPs even within a genome that can happen. But you're right, there is some sort of correlation somewhere, and I guess I can't talk too in depth about it, but like in a sample like this, I have single-celled organisms, and then I have organisms that have, nematodes might have 1,000 cells, or rotifers might have 1,000 cells, So it's hard to say that, I don't know. Part of me thinks that maybe sequencing technical replicates too might have helped, to sequence additional samples, DNA replicates from the same sample. And I kind of wish I would have done that now in hindsight.
Beth, I think you hit the nail on the head when you mentioned the part about in the eukaryotic survey, you have organisms that range from single cell to many, 1,000 cells, or in some cases more. And that is one of the reasons that it makes it so difficult to really quantify. However, if you had an idea of a particular group, how difficult do you think it would be to pull out a group of like all the nematodes? You expect them to have, roughly a similar number of cells potentially, or at least be able to find in the data how many cells make up the different each different species on a generally known basis. And then if full genomes are sequenced, or if they know how many copies are in a genome, you could correlate that.
Yeah.
Probably wouldn't want to do it with your entire data set. I think that would be extremely cumbersome, but I do think for those phyla that are interesting, or for those lower taxonomic groups that are really interesting, you could pull a subset out, do a little bit of database searching and see what kind of information can you glean and apply a model, have someone help create a model to simulate. So, and I do believe that there have been some publications that have done similar things in working out the methods, which brings me to another question. You tried a lot of different primer sets. Can you tell us a little bit about your exploration with primer sets and choosing those particular ones that you settled on?
Yeah, so I did a literature review on some different next-gen platforms where they had used these different primers for the variable regions for 18S, of course, because with any next-gen platform, you're limited to size. And it was largely decided that those V4 regions and V9 regions would be best. A couple of publications even said like the V2 region. So I just ordered several primers for those different variable regions and sequenced them, and to see which, and I just looked and saw which one gave me the best resolution. With that V9 region, I probably chose the wrong primer because it amplified a bunch of bacteria, whereas my other primers didn't, and probably just had something to do with the reverse primer. I think if I would have chose a different reverse primer, it probably might have eliminated those bacteria, but that's, I guess, always an issue is amplifying bacteria because they're sneaky, But yeah, so I just sequenced a bunch of different primer sets and analyzed them and looked at my OTU output to see what sort of matches I got from those primers. And actually ended up settling on what the majority of the literature said, so yeah. And I tried CO1 as well, but I didn't get it to amplify. I kind of wish I would have just sequenced it anyways, because sometimes those amplicons don't show up on the gel. But I ended up just kicking it out, but I kind of wish I would have kept that CO1. That would have been helpful too.
You're actually kind of limited because, you know, there's a history behind some of these genes. And as it turns out, you know, because prior to the technology for sequencing DNA, that people sequenced RNA. And so there was a lot of that data there, and that provided the information to create primers that then people started using. And so if you go to GenBank, you know, the ribosomal RNA is the best represented,
Yeah.
you know, in terms of what's in GenBank. And so you're kind of stuck with that in a sense. And yeah, it's you'd think everything's in GenBank. It's not even close. So.
Well, and there's other databases, but like GenBank is pretty much the easiest to use, you know. But
so sequencing ribosomal DNA can be kind of notorious for getting hits from contamination from your dog or from people. Did you find anything that was just like stood out that was either you or like in the environment, because it is a human, you know, humans are in there?
So I had one OTU in all of my samples, surface samples and underground samples, that matched a ambiguous tetrapod. And it was like, when I looked at it in GenBank, it was like 100% match to humans, to dolphins, to gorillas, to dogs. It was just like every tetrapod that there is. So it could have been contamination for sure. Yeah. It was some strange, like maybe conserved region or something of 18S. But yeah, I did, get a weird match. Yeah.
So one question that often arises, and I haven't heard anybody ask it yet, you've kind of addressed it because you sequenced surface organisms to do a comparison. But because of the fact that you were looking at biofilms, and because this area has been mined for, you know, it was mined for 100 and some years, plus another, you know, decade of people being underground to work it as a laboratory facility. Can you address the ecological significance of the diversity, its origins? You know, are there portions that are probably endemic to that underground site, others that have been introduced, and what might those interactions be?
Yeah, so as far as the actual origin, you know, I can speculate on where it came from, but yeah, because there's so much anthropogenic interference, can't just say, oh, these are endemic to a mile underground. There has been other research into deep underground areas where there are supposedly these microscopic invertebrates living happily in the fractures in the rocks and in the underground water. Yeah, so at this time, we can't say like if they actually came from the water source or from humans, came down there, tracked in on someone's shoe or whatever.
Or in donkey poo, considering that
Yeah,
before the rail cars came down, they used donkeys to move all that.
Yeah.
But over time, because you're in areas that have really been untouched for almost 2 decades, other than, you know, a few people going in now and then, some of those interactions I would think would become unique.
Yeah.
Because you've got things that were probably introduced and may have been introduced and reintroduced and then left alone for a while, interacting with those things that are endemic. So you really create very unique and very possibly interactions that wouldn't have existed normally.
Yeah.
And so you have the potential for some new ecological relationships, some new relationships between species, some syntrophy that might happen because their energy is limited down there, really limited. And so you have to have organisms down there that are capable of living off the rock and the water that's down there in order to get things started, at least.
Yeah, and then there's got to be toxic compounds in some of those biofilms that the microbes excrete, and I wonder, how do some of those eukaryotes deal with those sulfates or that high iron concentrations in those biofilms? How do they survive that? Yeah.
So those sites that you may have mentioned this, but those sites that you took samples from, was that area flooded?
Yeah, so it
prior to
flooded about
dewatering?
200 feet past the 4850. If my memory serves correctly, the 4850 did flood, so yeah.
That's
Yep,
kind
it
of
was.
interesting to think about too.
Yeah.
Oh, actually, Beth, in any of those papers about the Helicephalus, or what, the devil nematode basically in South
Okay.
Africa, in the genome, is there anything that might suggest that it can do anaerobic respiration,
Oh,
maybe using
yeah.
sulfate or something weird?
Good question. I did read that publication because it was relatively new, but I don't remember off the top of my head what else they discovered in that genome. But yeah, that is a good question. I think it did have something to do with maybe being facultatively anaerobic, that nematode, yeah.
Yeah, so it might have survived, even that flooding episode, perhaps.
Yeah. Oh, and then even that reminds me, so from Borgoni's previous publications, he did a bunch of different experiments, but he did determine that some of those nematodes just need very minimal oxygen to survive, some of those species that he saw down there. And I don't know if that goes for nematodes in general, maybe I guess I don't know a ton about them, but yeah.
Hey, just a comment. I think there's a 60 minutes episode on that.
Yeah. I couldn't find it on YouTube.
Oh, it's not there. It's
No,
fairly
there's
new.
a snippet of it, but I couldn't find the whole thing. Yeah.
It's pretty interesting
Yeah.
how they get down there.
Yeah. It's a lot harder for them to go, from Belgium to South Africa than for me to hop in my car and drive 25 minutes to lead. Yeah. Pretty lucky. All right. Do we have anybody left here? Have any more questions?
I have a quick question. I might have guessed that fungi would be more common in your samples, especially just because how I think about fungi. Also, Cindy can't stop talking about fungi and how awesome they are. So how do you interpret just sort of their absence? Well, not absence, but not prominence.
Yeah. So I sequenced that ITS2 gene, and I did get 3 OTU matches. That was it, that were able to be classified at 97% a taxonomy match to 2 different classes of fungi, and that was it. And it actually wasn't very many sequences either. So I have thought about that, possibly competition from bacteria, if these bacteria are producing antibiotic substances, and possibly that's a reason that the fungi aren't very prevalent, or just competition from them as well. It seems like in a biofilm there would be lots of dead organic matter. So you'd think, yeah, why aren't there more fungi or something down there? Yeah.
In terrestrial caves, are fungi common?
Ooh, I'm not sure. That's a good question. Yeah.
In terrestrial caves, they occasionally find what are called they're basically the dark fungi that can live. They're demaciaceous. They're dark colored. They tend to be able to metabolize chemo types. They're chemoautotrophic and chemoheterotrophic, or not chemoautotrophic, chemoheterotrophic. And so they don't need a lot of organic matter. They can actually survive off of limited organic matter, but they still need some. And there really is, other than in those biofilms themselves, there's not a lot of organic matter. Where you see the fungi underground is mostly on the timbers, the man-made structures.
Yeah. Yeah, and So yeah, a lot of fungi on the organic matter down there, everywhere. Possibly, what did we thought? We thought we even saw a slime mold one time.
Yeah.
Yeah.
Hmm.
And a man eating penicillin shortly after they drained penicillium. Shortly after they drained it, I have a photograph of an entire 9-foot by 9-foot crib completely engulfed in a penicillium, and it expanded out behind it on the ground. It was pretty monstrous.
Yeah, lots of stuff.
Stuff of nightmares.
Yeah.
Any more questions out there? Sounds like everybody's satisfied, so I guess clap one more time. Yay.
Thank you.
Good job. And I think with that, then we'll take a little break while everybody logs off, and then we'll just convene back in 5 minutes or so, and
Okay.
with the committee. Does that sound okay?
Yep. Sounds good.